Thank you so much for joining this uh, makeup class. Actually, we missed several classes. Today, I shall discuss on genome editing, which we started but didn't complete the whole lecture. So please notice me where I stopped in my last lecture. Is it visible now?
Yes, sir.
Okay,
জি স্যার দেখা যাচ্ছে স্যার।
very good. Then please tell me where actually I stopped in my last lecture. Possibly I discussed the methods, three types of,
Sir, it has been discussed, sir. Please go
uh,
forward.
yeah, three types of genome editing. Generally, we group them in three groups, SDN1, SDN2, and SDN3. And I discussed site-directed nuclease 1 and 2. They are non-transgenic, but SDN3 are transgenic. This is why the resultant organism can be considered as GMO. But SDN1 and SDN2, in case of SDN1, just to cut the uh, double-stranded DNA, and then DNA automatically repair, and here an indel deletion of one nucleotide or maybe more if can be happened. And this type of editing is non-transgenic. And here, if the target gene uh, is an important gene which carries a negative uh, traits, that is, for example, disease susceptibility, then even deleting one nucleotide can inactivate the expression of the gene. So this is a very simple way. But in case of SDN2, here we can correct a wrong DNA sequence, maybe mutated somehow. So you can keep a template of corrected sequence, and then it will fill up, and the error can be repaired or edited. And in this case, no new gene is added. Rather, uh, the error in the DNA sequence or any gene can be corrected. But here you can put a new gene uh, which you want to insert, like a BT uh, gene, or like cryo-1AC gene for making the BT brinjal, or like that, you can do it. So SDN1 and SDN2 are considered non-transgenic, and they are heritable. And most of the country, uh, those who have already allowed green pass to the gene edited product, they considered them like classical mutation breeding. So without any strict biosafety guideline, you can go to the field. In Bangladesh, we also approved, our government approved a standard operating procedure where SDN1 and SDN2 approaches you can use for developing uh, improved crop varieties. And if you can prove that there is no transgene in the uh, breeding line, then edited line, you can go to the field directly. We have a committee for approval of this type of gene edited product. So major gene editing technology I already discussed. There are three uh, major technologies have been discovered: zinc finger nuclease, TALENs, transcription activator like effector nucleases, and CRISPR-Cas methods. And CRISPR-Cas is the latest one, discovered by Emmanuel Charpentier and Jennifer Doudna group, and they received Nobel Prize in 2020 in chemistry because this method is the smartest gene editing method. By using this method, you can edit the genetic code more easily than the other method. And it is the paper published in 2012 in Weekly Science, which is the revolutionary paper, a programmable dual RNA guided DNA endonuclease in adaptive bacterial immunity. Adaptive bacterial immunity means CRISPR-Cas system is a natural uh, adaptive system in bacteria and prokaryotes, which was first detected in 1987 by Dr. Ishino in Japan, and then from 1997 to uh, 2011, scientists confirmed that the presence of CRISPR-Cas system in the bacteria is nothing but an immune system to defend from the attack of viruses, because virus uh, can attack the bacteria and prokaryote to make the copy of their DNA or RNA. And through the evolutionary process, uh, bacteria somehow cut the DNA from the virus and put into their own DNA, and also evolve an enzyme, Cas enzyme, CRISPR-associated enzyme, and it uh, evolved as a molecular scissors. So next time, if similar uh, virus attack them, they can cut the DNA and inactivate the virus. So CRISPR already, you know, that clustered regularly interspaced short palindromic repeats. That means the uh, cluster of the nucleotides present after certain distance, and if you reverse them, you can read similarly. This is why they are called palindromic. So a genome editing technique, CRISPR is now the most popular genome editing technique that targets a specific section of DNA and makes a precise cut or break at the target site, and can do at least one of two things. The first one makes a gene non-functional if it is carrying negative uh, or bad gene or detrimental gene, undesirable gene in an organism, you can make it non-functional. Just I mentioned before, the SDN1 system you can use here. Replace one version of a gene with another. That means you can correct it. This is also SDN2, and they are transgenic. Now the question is, why CRISPR-Cas system or technology is superior? It is superior because the previous zinc finger nuclease or TALEN, uh, they are cumbersome. Why? Because both methods require sophisticated protein digestion, synthesis, and validation. 3D protein structure and their digestion synthesis is really complicated. But on the other hand, CRISPR-Cas system or CRISPR-Cas9 system, 9 is more popular, but there are many CRISPR-associated protein or enzyme have already been discovered in eukaryotes. CHIP relies on single guide RNA to target site specificity, can be readily designed based on genome of choices and in multiplex fashion, in which several genomic loci can be perturbed or edited at the same time. This is a very fascinating thing. That means you can edit several gene or several places in the DNA at a time, just designing the several guide RNA targeting the specific genetic gene sequence. That means you can do multiplex genome editing. System create at a time. System create stable and heritable mutation. That means the genome edited organism, they are heritable, not like hybrid. So you can offer the seed to the farmers and they can keep their own seed and go on like mutation breeding. Transgene-free plants in a few generations, 2 or 3 generations. If you do, then even CRISPR gene can aborted and the plants become uh, transgene-free, only the edited one. Edited plants variety can be used for plant breeding. You can make lots of changes in the DNA by designing the guide RNA and make lots of diversity or variations, and those plants can be used for breeding as uh, uh, like a variant. When you create, then you can transfer the phenotype through the classical breeding also, or molecular breeding. New set of CRISPR-Cas system like Cas13, Cas12, and lots of other new systems are coming, and they have some sort of advantages for the editing. CRISPR is unique because it derived from a natural process I mentioned before, found in bacteria and archaea to protect themselves from the viruses, and targets genes for editing and regulating. And third point is comparable to Photoshop. It is you can as we can edit the photo by using Photoshop. It is like that. So you can change any code of the genetic code of any organism. And here the simple process, what you need to do, the yellow one, the guide RNA. That is this guide RNA, single stranded one, you design in a way to target a specific DNA sequence where you want to edit. Then you make a complex with the Cas9 enzyme, and then this guide RNA, read the complementary sequence in the DNA in the cell, and uh, when it finds the right sequence complementary to the guide RNA, so it is like an address, then the Cas9 cut the double-stranded DNA like this, and then the mutation taken place. So generally, some part can be deleted. Even you can make several guide RNA and completely remove a particular sequence from the DNA. Or if you put a corrected sequence, and then you can make a correction. So really interesting. And here is the illustration I previously discussed. This one. This one is the simplest. What you can do, you can cut the double strand by using CRISPR, Cas9 and guide RNA designing to target the double-stranded, your gene of interest, and then it cut the double strand. After cutting the double strand, several things can happen. I mentioned already indel mutation, only one nucleotide can be deleted. On the other hand, you can cut in, two guide RNA and a fragment can be removed. So when DNA has a repair and maintenance system, so if you remove this part, then the joins together. As a result, a bad gene completely you can remove. And on the other hand, you can also replace a, bad sequence, undesirable sequence, if you put the complementary sequence. So this part is called a non-homologous and joining repair pathway. On the other hand, here a homology-directed recombination pathway. Double strand cut is similar, but where you can do, you can also, insert a gene, new gene into the DNA sequence, or even small correction can be done as by using double-stranded complementary DNA sequence. So this is very important and very, uh, clear-cut principle of various kinds of genome editing and manipulations by the CRISPR-Cas system. And in the plant genome editing, mostly non-homologous end joining repair pathway are being utilized for uh, developing the new variety. But this HDR is also in research hugely being used, but due to the GMO concern, uh, this is not been practically used, but huge research are going on for making the HDR pathway mediated genome editing. In that case, even chromosome engineering can be done, and there are lots of basic works being done. And here you can see how the process is done. The single guide RNA, if you know your gene of interest, then you can design like primer, the single guide RNA, or you can ask the company to design the single guide RNA. And Cas9 nuclease is available in the market, so you can buy them because it is a natural enzyme. So these two makes a complex Cas9 or Cas, guide RNA complex. And then this is the, DNA of the cell you want to edit. So what will happen, you can see now the Cas9 and guide RNA guide to find the target sequence, the red one, and then it stops here. So address is found. Then the uh, what happened? The Cas9, this enzyme, it nuclease cut the double-stranded DNA, and then here you can see very interestingly several nucleotides around beside the cut point, removed, and in the DNA repair mechanism, sometimes it happen. Here you can see the edited gene, that is three nucleotides being changed. So by using the construct of, targeted CRISPR-Cas system targeting a specific gene, you can edit like this way. And it is really very, very useful for, developing disease resistance, or even in case of fisheries, for example, red sea bream or tiger pufferfish in Japan, both commercialized the negative regulator of the muscle growth, myostatin protein regulating gene disrupted by this method, and 40% or even 50% muscle growth increased. But the organisms are non-transgenic. They are being commercialized in Japan. So there is another editing system called base editing. Base editing is a genome editing technique that changes a single base in the DNA, not the nucleotide. Nucleotide means base, phosphate, and deoxyribose sugar. So in DNA, RNA, only a single base, A, T, G, C, anyone can be edited if it is wrong. So it's a precise way to introduce single nucleotide variant, that is SNPs, into the cells. In 2016, David Lewis' team at Harvard University developed a base editing system. How does it work? A CRISPR guide RNA molecule binds to a target sequence. A base editor, which is a fusion of a nucleobase deaminase enzyme and a catalytically disabled nuclease, converts a base or a base pair. Now, that is very quite interesting. CRISPR-Cas system, when it develops very rapidly, the improvement of the methods, we witnessed how faster it is evolved. And base editing is also one kind of CRISPR-Cas system. The edit is completed during the DNA replication by the mismatch repair and base excision repair mechanism, because in the uh, cellular system, base editing or nucleotide editing system, nucleotide or base repair system is present. So if you can somehow, target a base or nucleotide and cut that one cell in its own inherent mechanism can repair, and the base can be changed. Here, uh, uh, base editing can silence disease-causing gene or activate specific genes. It has the potential to treat many genetic diseases which are often caused by single nucleotide variants. For example, even um, uh, cancer disease, blood cancer, one of the patient for the first time in UK, Alisa, she had no hope because all the treatments were failed. In that case, a single base editing resulted a complete cure of the cancer. But bases editing can be two types: cytosine base editor. Another one is adenine base editor. Change a cytosine guanine pairing to a thymine adenine pairing. On the other hand, changes an adenine thymine pairing to a guanine cytosine pairing also can be done. So specifically, specific base pair can be changed here. Base editing is demonstrated as CBE, cytosine base editor, and here adenine base editor, how it can be done. Here also guide RNA, and instead of Cas9 here, Cas9, engineered Cas enzyme is used, and some other cytidine amine, deaminase or deoxyadenosine deaminase, this enzyme used to make the construct, and then the target, the base is edited. The process is a bit complex, but I tried to show you the simple way how a specific base can be edited. Another editing method is called prime editing. The name implicates that it is a bit advanced method. In that case, double-strand breaks is not necessary. Prime editors do not require double-strand DNA breaks, can make virtually any substitution, small insertion or small deletion within the DNA of living cells. So prime editing is one of the most advanced editing system in the genome editing. Prime editing minimally requires a programmable nickase fused to a polymerase enzyme. That is, several enzymes are required to make the construct, and an extended guide RNA that both specifies the target site and templates and the desired genome to edit. So prime editors offer an unusual combination of versatility, specificity, and precision. So three unique properties of the prime editing are versatility. It is versatile. You can do lots of things, lots of changes, and specificity, more specific, less off target, and precision obviously is very, very unique in case of prime editing. How prime editing works here, here you can see the illustration. Prime editor complex initiates search for the target DNA. This one, the complex that is construct you need to design, and then it searches for the target DNA, and then here you can see it, find it, and then cut the single strand, not the double strand. Here, single strand, and then did the editing, and finally, specific editing being done very precisely, and replace prime editor complex copies in corrective DNA sequence. Here, for example, there are some errors, so you can correct it. So finally, gene corrected. So single, for example, single nucleotide polymorphism, sometimes a big problem. For example, sickle cell anemia disease can be edited by the this prime editing. So prime editing here, I showed the major steps. Please go through. Uh, no double stand breaking. And here, instead of only Cas9, that is engineered Cas9 enzyme is used, and prime editing guide RNA targeting the your specific site of the DNA you are going to edit, and it guided by 20 nucleotide sequence within the prime editing guide RNA. So the guide RNA size is around 20 nucleotide. And after flap equilibrium, you see after flap equilibrium, it can correct like this way. So it can do lots of, diverse things. And there are lots of, genome editing methods have already been established. For example, target knockout, I discussed before. This is simplified one, Cas9 enzyme. So to do knockout, you can design a single guide RNA or double guide RNA, two guide RNA to specifically silence or knockout a specific segment. Base editor, prime editor, or expression control. Even by using advanced method, you can, without any cutting the double strand, you can even control the expression of the gene or epigenetic modification. Epigenetic modification means if there is ATGC, the sequence same, but if in the sugar hydroxyl group is replaced by methyl. In that case, even you can correct it. Inducible gene expression, even effector level, you can modify. Repressor, you can use DNA methyl transferase or, degron. Lots of things now emerging to engineer the or bioengineer the cell or the RNA or DNA. So in CRISPR-Cas system, when Jennifer and Emmanuel published the paper, they published in 2012. Later, in 2014, it was a craze. People asked, how can we use CRISPR-Cas9 system? So they thought this method could revolutionize in agriculture, crops, animals, genome editing, human gene therapy, the most important target for, editing the mutation that result in cancer or even diabetes or other genetic diseases can be done very precisely. Screen for drug target uh, because it can silence the gene, so you can target the drug molecule target or ecological vector control like mosquito sterilization, viral gene disruption, pathogen gene disruption, and even synthetic biology, pathway engineering, because you can do lots of basic work and programmable RNA targeting. And practically, CRISPR-Cas system has hugely been used to do the basic research, because by this method, precisely you can inactivate or silence a particular gene and confirm its function by developing the mutant. So in case of CRISPR, first the job is design, and then the next job is deliver. To deliver the job, you need to do transformation. So CRISPR-Cas system, still a big job is you need to use either agrobacterium mediated transformation or gene gun or other nanoparticles to deliver the construct to the the DNA of your cell to allow editing. So people are now trying hard to get the CRISPR expressed. That means without any, uh, uh, you know, uh, agrobacterium or gene gun directly, yeah, in vivo condition like in planta, without tissue culture or other method, how can editing be done? So it is progressing and there are significant progress. And then after editing, the next job is to confirm the edits by using sequencing, PCR, even phenotyping analysis you can do. For example, if you want to inactivate a salinity susceptible gene, and then you can grow the plant or organism in the saline condition. If you see they are growing, then you can confirm that tentatively they are edited. But the precise one is the sequencing of that gene and see whether or where it is edited, how many nucleotides are edited. So CRISPR is faster method. Time required for the genome editing of a plant is very short. Even within few weeks, you can do the editing. And next, few generation in the spread breeding system, you can do. So within a couple of years, you can even get the fully stable edited line. One of the stunning finding of CRISPR-Cas system is that wild tomato over last 10,000 years through the different breeding and selection process, we have now high yielding tomato plants. But high yielding tomato plants are susceptible to many biotic and abiotic stresses. But the scientists sequence the cultivated, domesticated tomato as well as the wild tomato. They found that some of the genes are domesticated traits like a compact plant architecture, synchronizing fruit ripening, day length insensitivity, and large fruit size, increased vitamin C level, increased fruit lycopene level, fruit size and weight, fruit number, and fruit shape. These are the major genes have been, changed from the wild to the domesticated one over 10,000 years of, our breeding practices. And now we have this tomato. Scientists targeted, all the genes by using multiplex genome editing CRISPR-Cas system. And then they found that within a few generations, de novo domestication, that is a very fast domestication by using genome editing can be done. And in that case, the major advantages would be biotic and abiotic stress tolerance will remain here. So this type of editing, if commercially being utilized, then there will be, climate resilient and climate smart crop. Similarly, in case of rice, wild rice very poorly yield and still exist in the nature. And the cultivated rice, several scientists edited these genes and found that it is possible. So more precision can help us to, orphan crop or, wild relatives, you can edit and make them commercially applicable. And obviously they will be highly tolerant to the adverse, uh, environmental condition. That is, they will be tolerant to biotic and abiotic stresses. And people in genome editing, for example, this is a very interesting example. Powdery mildew resistant tomato, even wheat developed because amylolocus are responsible for the susceptibility of the powdery mildew disease. So once here you can see they edited these nucleotides, no transgene here found. Plants became immune. In case of wheat, Kaixia Gao in China, they also successfully edited the disease, wheat plant and the powdery mildew resistant wheat. Here, tomato being shown. So in that case, there is a risk of, pleiotropic effect. If this gene is connected to the yield or other good trait, then this type of editing may result a problem. In case of wheat editing, for powdery mildew resistance, Gao first found some yield compromise in the edited lines. Then they edited further to improve the yield, and they minimized it. So I am at the end of this talk for uh, a little more discussion. What we can do, disease-resistant plant we can develop by using genome editing, removal of deleterious alleles from the genome, yield improvement. There are lots of literature. Yield improvement can be possible. Input use efficiency, that is nutrient use efficiency, can be increased, or water use efficiency can be increased. So lots of literature you can find. And recently, a big advancement is taken place. For example, here you can see in the grafting system, and the upper part is called scion, and the lower part is called rootstock. So if you make the CRISPR-Cas, construct, if you make a transgenic of rootstock, and if you put a scion on it, interestingly, through the vascular system, CRISPR construct can go to the up and can edit the plant. So if you buy the rootstock transgenic containing the CRISPR, construct, then you can do, editing by grafting only. So it is really interesting. That means transgene-free plant you can get uh, from the seed, and then you grow and you can select it. So this is really, even maybe in a hydroponic system, if you put the CRISPR, and then if plant uptake not yet been done, in case of rice, if it can be done, that would be fantastic. So several commercial crops have already been available, edited one, soybean, camelina, oil producing crop, Sitaria, green Bristol grass, waxy corn, white button mushroom, and in Japan, tiger pufferfish, red sea bream, and genome edited GABA enriched tomato. So these are the some examples. I am going to stop here. So if you have any question, we can discuss. And in our case, we also did some of the smart job, especially the as a guide RNA can specifically target a specific DNA segment. We developed the diagnostic kit. So I will take a little pause and allow you to ask some question, and then I will show few slides to complete this lecture. So it is your time. You can ask some question. Anyone just unmute and then ask your question.
Sir, thank you for your nice presentation, sir. Sir, I have a question. My question is, sir, how can we create a complex of single-guided RNA and Cas9?
Thank you. This is not so big deal, because first you need to know which gene you are going to edit. So that gene must be characterized, must be cloned precisely. If you know the gene, then you can and use the software like we design the primer. So guide RNA you can design, and then you can order the guide RNA as well as Cas9 enzyme to the company. And they are actually, especially Cas9 is patented. Even CRISPR system is basically patented. For research, it is no problem. So you can order the guide RNA and Cas9, and then you can make the construct. And that construct you can use for the editing. Editing
Thank
of
you,
your
sir.
cell. Thank you. And next,
Sir, another question, sir. Sir,
yes.
yes, sir, how we insert this construct into the plant?
Yes, in that case, like GMO production, you need a method for transformation. For example, agrobacterium mediated transformation you can use, or you can use the biolistic gene gun. In that case, if you want to do in case of plant, you need to remove the cell wall and get the protoplast. You know the protoplast. When we remove the cell wall from the plant cell, then it is called protoplast, only membrane-bound cell. So by using biolistic gene gun, CRISPR construct coated with the gold or platinum, you can, by force, you can transform the cell, and then through the tissue culture, you can get the plantlet, and then you do the screening and check which plantlets from the edited protoplast are really showing the phenotype that you are desiring. So the rest of the job is like genetically engineered plant. We do the tissue culture and then screening, and you can also do the sequencing PCR to confirm whether editing was done. Any more? Sujan? Sujan there? Any question? Because in fish, especially tiger puffer fish and red sea bream in Japan, they are very famous and delicate for the sushi. And in both cases, the muscle regulating protein, myostatin, were edited. And this is why muscle production increased. Any question? Ratna and Jannat?
স্যার প্লান্টে ইনসার্ট করার জন্য ডিফারেন্ট মেথড আছে আমরা জানি, কিন্তু আমরা যদি স্যার লাইভ হিউম্যান বা লাইভ ফিশের ক্ষেত্রে আমরা ট্রানজিনটাকে ইনসার্ট করতে চাই বা জিনোম এডিটিং করতে চাই, সেক্ষেত্রে স্যার মেথড গুলা কি হবে?
Yes, this is a very important question. People have started, editing a particular specific nucleotide, that is single nucleotide in case of the sickle cell disease. A single nucleotide error is now correcting by using genome editing. The process for human or the whole organism is a bit complicated, but in case of plants, for example, some scientists have developed implanter method. You spray the construct on, germinating seed or like that. So then, it can be done. Even grafted, scion can be used. But in case of animal and, other organism, the smartest idea to edit at the germline stage. Germline means embryonic stage. Then it is very, very easy and precise. So if you edit the pluripotent embryo, a particular gene, for example, in case of human embryo, the Chinese scientist, he edited the after in vitro fertilization, the embryo edited, and two babies, test tube babies, Nana and Lulu, they are free from the CCR5 gene, because CCR5 gene is responsible for a penetration of HIV virus, because both of the parents are, HIV patient. So the couple wishes to have baby without any risk of HIV, AIDS disease. So it is possible. So the smartest idea is the whole organism, especially in case of human, for example, sickle cell disease or any other gene therapy, still very complicated. There is a risk of, off-target editing. Through the vascular system, it is done, cell edited, and for example, blood transfusion or other ways, even stem cell therapy can be used, but obviously complicated. But at embryonic level, in case of fish or tiger puffer fish, is very, very smart way. And in case of plant, the smartest way is to do the at cellular level and do the tissue culture and get the a plantlet and the whole plant. Any more question?
No, sir.
Okay. Thank you. Then I will show you a little more information today. This is, I mentioned that amylolocus are responsible for the susceptibility of wheat to the powdery mildew disease. And the Chinese scientist Kai Gao group, they edited here, you can see the amylolocus edited and plant became the immune to the uh, powdery mildew disease. And in case of wheat and soybean, the gene edited crop being released in China also. And this is another example, vegetable edited by uh, CRISPR, people eating. And this is the, uh, stunning one. And I think most advanced one, sickle cell disease therapies using the CRISPR-Cas system. Here you can see only single cell, single nucleotide, uh, mutation, red blood cell became sickle shape and they suffer from the anemia. So in that case, correcting one nucleotide resulting the a complete cure of the disease. And in US, they started, the editing in 2023, almost 2 years, and many patients recovered. And this is another example. I told you, He Jiankui, he edited the human, uh, the human germline, that is embryo, after in vitro fertilization of the sperm and ovum of a couple. And they got two babies, Nana and Lulu. They are free from the AIDS. And one case, only in case of cancer, Alisa in UK, she was the fourth stage of blood cancer, and all the treatment failed. In that case, base editing cured her. And you can ask why, in case of blood cancer, these technology are not being spread, because still there is a risk. And in case of human application of any technology, it should be 100%, specific and accurate. And the accuracy is still a challenge. So CRISPR-Cas system can be used for many purposes. One of the biggest, robust use can be the disease resistance. Disease resistant or disease diagnosis. Disease resistance by using S gene editing, we can do it. And disease diagnosis is very important. I can show you our method. Oh, just a moment. Yes, we, first developed a, PCR method by using the genome-specific primer. We discovered MOT 6098 and 6099 in the genome of Magna Porte or IgTTCAM, because there were no genome-specific primer. And then first in the PCR, they were confirmed, and then another method called loop-mediated isothermal amplification method for detection of the specific primer, DNA fragment in the sample. Here, some instrumental facilities are required for temperature and others, but PCR, it is confirmed. But in collaboration with Hao Jiang and Gu Liang Wang, by using CRISPR method, this type of strip you have seen in our lab being developed. So in that case, the guide RNA Cas12 enzyme target this nucleic acid fragment in the MOT, and then it develops two band and single band. And here, instead of PCR, we use the RPA to amplify, uh, 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 you know, the tiny DNA in the sample, and then this type of PCR strip. We use Cas12 enzyme and many other, visualizing agent to visualize it. So this method is very interesting. I would like to show this one, how to say, yes, here you can see the major steps. That is, you just crush the plant sample and in the buffer. Buffer is provided in the kit, and then you take out the DNA in a strip, and the strip again put into the buffer, and here you put the RPA. RPA, amplify the DNA and amplified DNA. Here there is a construct we build with the biotin, programmable guide RNA, and dead-Cas9, and then if you and mix here, and if you put one drop here, then you can find two-band if there is a disease. So this is the smartest diagnostic kit developed in our laboratory, and you can use this platform for detecting any nucleic acid, even it may be the human disease or animal disease or fish disease. And we are now uh, trying to get it more stable and cost effective, and also to utilize this method for fish and other disease detection. So I should stop here, and later maybe other class I shall discuss more. So your midterm exam on 27th will be up to this lecture. So if you have any question, we can discuss a little more. Do anyone have any question? So 3 p.m. on 27th, you will have the midterm exam, right? Raihan?
Yes, sir.
Okay. So uh, I offered you several lecture, tissue culture and transgenesis, GMO, how genetic engineering can be done by using agrobacterium and gene gun and other method, and the challenges associated with the uh, bioengineering of plants and other organisms already discussed. And GMO not only available in plants, even fishes and other organisms, GMO are now available. But in case of GMO, there is a challenge of the biosafety process, very long process, and also some people are skeptic policymakers about the GMO. So gene editing is an advanced bioengineering technology, which has an added advantage that you can get the transgene-free, bioengineered, improved organisms, as Japanese are using gamma aminobutyric acid and this tomato, red sea bream, high muscle production, or tiger puffer fish, and in America, or the abiotic stress tolerant soybean, and browning resistant mushroom, and high oil content, good fatty acid content, camelina. So several products are now available in the market. In China, they have the soybean, they have the wheat, and many more are coming in the pipeline. But the biggest thing is that science is of genome editing is explosive. If you check the literature of genome editing, it is exponentially expanding. I expect that one day the genome editing technology would be like a PCR method in every laboratory. It is not only developing a product, but also dissecting the biology of an organism, as it can silence, inactivate the gene, and can do lots of multiplexing, can create the biodiversity, and many more that we are imagining. Even chromosome engineering can be done. So to understand the complexity in biological system, even the pathway or system biology, genome editing would be a fantastic tool, and it is already here. And another advantage is that it is an adaptive immune system from the nature. We are getting this engineering tool. So, any question? In Bangladesh, if I ask you which area you think is the most important for application of genome editing, can anyone share your thoughts? Sujan?
Yes, sir. Uh, Actually, plant is the best area for the genome editing because actually there is regulation and regulation and policy is very low restriction in here, whereas in fish or human side is very restricted in regulation. So,
Yes. Yes. In case of plant, actually, no ethical and strict guidelines for at least doing the research. And even to go to the field, we have SOP already in place. You are right. But in other areas, I'm expecting fisheries and animal science, they will start the genome editing because other GMO development and other things are very complicated. But in now, genome editing can easily be done as some of the cases are already been successful in some countries. Yeah.
yeah,
Fisheries may be not so complicated like, other animals, large animals, or even the human is obviously highly restricted. For example, editing at the embryo, I told you, is
yeah,
also highly restricted because otherwise people may desire a designer baby. So
yeah,
if there is any wrong, then the baby may be uh, very, very strange, thing. So off target, in case of plant, even fish would not be so, serious. You can kill, but human baby, you cannot kill through
yeah,
the process. There
yeah,
are
sir.
strict ethical issues.
Right, ethical issues.
Any thought from other? Jannat, Raihan, and Ratna?
No, sir.
Yes, the non-transgenic genome edited product is like your mutation breeding doing at your institute. And the thing is here, the precision and, faster the process. These two things differ from the classical mutation breeding. Ratna, any question or
No, sir.
comment? Comment? Have you enjoyed the talk?
Yes, sir.
Okay, then good luck and good night. I will share the, a lecture if it is recorded well and try to upload in the YouTube, and you can listen later on also. Thank you so much. Good night.
Thank you.